Hello, welcome to Statistical Analysis of Ecological Communities, a new course for spring 2021 at Texas Tech University. My name's Nancy McIntyre and I'll be the instructor for the class. I'm a professor in the Department of Biological Sciences here at Texas Tech, and I developed this course to fill an empty niche at Tech. There are several courses on ecological data analysis that are offered in the Departments of Biological Sciences or NRM. And there are also classes that use R for analysis. But there wasn't a class that was focused on biodiversity data. And so that's the focus of this class. So in this video, I'm going to give you an introduction to the course structure, and then I'm going to talk about what ecological communities are, why they're important things to study, why quantification is needed to study them, and what kinds of data we need in order to perform these kinds of analyses. This link is the URL for the syllabus for this class. I sent everyone an email with this link in it. Please be sure to bookmark this site because you'll be accessing it every week. At the bottom is a screen capture of what the top of the web page looks like. So this is where you'll find the online notes and videos for each week, the data files that you'll be using, the due dates for your assignments, my contact information, and the contact information for Iroro Tanshi, who is the teaching assistant for this class and to whom you will be turning in your assignments. The website also lists what technology you will need, the expected learning outcomes for this class, and the methods for assessing those ELOs. It will have information on Texas Tech's policies on academic integrity and academic dishonesty, and you'll find standard TTU info about what to do in case of illness, disability, or a conflict from a religious holy day. Finally, on the website, I also have a few links to some recommended books and to some tutorials on R. I haven't finished writing lectures or recording videos for this class yet, so be sure to check back often for updates. I've built this course around addressing 8 questions. First, what are ecological communities anyway? Why do we study them? And what kinds of data are needed? And that's the topic of today's lecture. Secondly, how do I describe the structure of a community? A community is a multi-species assemblage, and I'll get more into this in an upcoming slide. But basically, the aspects of a community involve the species richness, the number of species you have present, their evenness, in other words, how the distribution of individuals occurs across species, and those two things collectively define biodiversity, or diversity for short. And so we're going to talk about diversity indices, and we're also going to talk about abundance patterns. Third, how do I describe the structure of a taxonomic subset of a community? This is the specialty of Iroro's, and she'll be giving a lecture about assemblage structure along ecomorphological gradients. That's part of her dissertation research. Next, how do I summarize complex relationships and identify the most important patterns from a near infinite number of possible arrangements of multivariate data? In other words, we have large data sets that we'll be working with. How can we separate out the important bits from the noise? And this is the purview of a family of techniques that are collectively called ordination. And the remaining lectures of the course after this one will deal with various forms of ordination. Next, how do I winnow down a large number of correlated variables to an independent set of variables? This is the realm of principal components analysis, or PCA, and redundancy analysis, or RDA. I'll talk more about these things coming up. But basically, PCA allows you to detect any variable in your data set that explains species composition, and RDA allows you to detect the best explanatory variables in your data set. Next, how can I determine which environmental variables are associated with species occurrences at different sites? And we'll use 2 techniques for this. One is non-metric multidimensional scaling, or NMDS, and the other is canonical correspondence analysis, or CCA. NMDS is an unconstrained form of analysis that identifies patterns in a data set and the variables responsible for those patterns. CCA is a constrained form of ordination that identifies the effect of one data set on another related data set. How do I determine whether my data are grouped? And this is the purview of cluster analysis. If I have communities with a priori groupings, in other words, predefined groups, perhaps via cluster analysis, how do I determine whether or not those groups are in fact real, whether they're valid? And that's the purview of discriminant function analysis, or DFA, and there are many ways of doing so, and we will use a technique called CART, classification and regression trees. Now, I'll be approaching these questions from an ecologist perspective, not a statistician. I frankly don't have the expertise to get into the mathematical underpinnings of each one of these methods, nor all of the variations of each method, or even all of the different methods there are to address each of these 8 questions. My main objective in this course is to teach you currently best established practices or methods that are most heavily used in addressing these kinds of questions. And to do so, we will use the R computing environment. R is freeware. It has a steep learning curve, but is now perhaps the most heavily used statistical analysis package. There are many other software packages that can address these 8 questions, like Canico or PCORD, but no package covers as many types of analyses as R does, and those packages aren't free the way R is. So having experience and skills in R will make you a much more rigorous scientist. So let me clarify what this course is not and what it is instead. First, this will not be a deep dive into R. Instead, R is going to be used as a tool. There are other classes on campus that you can take to give you much greater mastery in R. My focus isn't going to be on R, it's going to be on the analyses and really the output of that analysis, of those analyses, and how you interpret that output. Secondly, this course is not designed to be a help session for problems you're encountering in analyses for your own research, especially the live sessions that we will have on Thursdays. I don't want you to co-opt that time to focus around your needs, which aren't relevant to the other members of the class. Instead, we'll be structured around only certain topics, and this is no substitute for consultation with a statistical expert who can get to know you and your data and your research objectives. And finally, this course is not comprehensive. It's an overview of the types of techniques that are used for the analysis of ecological communities. So by the end of the course, you'll have gained the following 5 things. First, you'll have an understanding and appreciation of ecological communities and the kinds of analyses that are performed on biodiversity data. You'll have knowledge about which statistical method or family of methods is needed to address a particular question. You'll have experience with the R computing environment, including RStudio and R Markdown. You'll have sample code that you can use in your own research. And finally, you'll have an ability to interpret the output of such analyses in the future. This course will not give you a comprehensive understanding of the statistical analysis of ecological communities, but it should enable you to conduct some analyses on your own, and it should empower you to explore other forms of analysis and feel comfortable in R to do so. So here's the course structure. This is technically a Tuesday-Thursday class. On Tuesdays, there will be an asynchronous session, meaning that I'll have a narrated PowerPoint video, like this one, a lecture, that I'll have posted on YouTube. These will be accompanied by detailed lecture notes in Word form on the course website. And those lecture notes will include samples of R code and example analyses. You will Use those because they will include your weekly assignments. You can do those at any time. That's what asynchronous means. On Thursdays, there will be a live session, live via Zoom. Now, this session is optional. It's really meant to be a question and answer kind of session. If you're having any questions or problems with the analyses, with the assignments, it's meant to be an informal kind of thing that where you can help each other and where you can collaborate and share tips and strategies and so forth. These Zoom links will be sent to course participants only. Please do not share them because we want to avoid Zoom bombing. The following Monday, that's when your assignment will be due, 8 a.m. to Aurora via email, and the instructions are on the course website. You will use R Markdown to create a Word document that you will email, and you'll do that for each week. So I recommend that you interact with the course materials in the following order. First, read the online notes and work through the exercises in them. Then watch the lecture video, the YouTube video, and finally, then do the assignment. In terms of assessment for this course, there are no tests. Your grade will be based strictly on the weekly homework assignments. There will be 13 of them. The assignments will be given in the course notes for each week, and what you'll do is you'll work through them. You'll create an R Markdown file in a Word document, and you will turn those in via email to Aurora at her email address no later than 8 a.m. the Monday after the week when that material was presented. And then please use the subject message of assignment on fill in the blank, like assignment on PCA or assignment on NMDS or whatever it is. There are instructions on the course website. Y'all, this is a graduate class, and therefore I'm treating it essentially as a pass-fail kind of class rather than one of more nitpicky traditional assessment. And what I mean by that is if you make an attempt at each assignment, even if you cannot get everything to work, you will receive full credit. The point of this class is not a grade. The point of this class is to provide you with experience that will lead to increased confidence in multivariate statistical analyses and in R, and to provide you with sample code that you'll be able to use in your own research in the future. So don't worry about point here, point there. This really is about, are you doing some work? If you get something wrong, for example, if you do an analysis in R and it's not the correct thing, or you can't get it to work, okay, well, here's what the real answer is. I will give you that in the corrected assignment back. So you'll be given the correct code or whatever the correct graph is or the correct interpretation of a graph so that you have that information. The important thing is the information that you get. And so I'm not going to treat this like a traditional class where we're doing tests and I'm counting off a point here, a point there when you get stuff wrong. That's not the point of this class. So I've got 3 important notes that I need to discuss with you all. The first of these is this is a new course. This is the first time that I will have taught a course entirely online. As you can see, I'm teaching out of my own home on an old laptop because my work desktop doesn't have a camera on it. I'm working from a PC platform, but I anticipate that some of you all will be using Macs. And we're dealing with R. And R is, let's call it finicky. So I expect there to be glitches in this class. Please inform me of any problems that you encounter right away so that they can be corrected. Secondly, the course materials are provided via a website rather than Blackboard because Blackboard is a TTU platform and it requires a TTU login. I developed and maintain a website that I use for all of my teaching materials in order to make these teaching materials more broadly available to the world. So, for example, there's a group of students from Malaysia who will be accessing these course materials this semester, and they will be essentially in the course even though they're not enrolled at Texas Tech. If I did everything by Blackboard, they wouldn't be able to access the materials. So if you have any, if you encounter any problems with the website, please let me know immediately. Finally, there is a disparity in experience with R. I sent you all a couple of questions a week or so ago asking about your experience with R and your experience with basic statistics. Everyone indicated that they had had a class in basic statistics, but the experience levels of R ranged from no experience to I've experienced it and I feel very comfortable with it. And so this disparity can make live sessions possibly uncomfortable for some people on the less experienced side of the spectrum if they feel like they're the only ones who aren't getting it. They may feel intimidated. Please don't feel intimidated. Frankly, some of you all have more experience with R and can do things that I can't. So in the live sessions, please don't hesitate to ask any questions. This course is about you all and you all gaining experience. So now that the course details are out of the way, let's get on to the course subject, ecological communities. So first off, what is an ecological community anyway? Ecological communities are multi-species assemblages. In other words, they're groups of multiple interacting species that are spatially defined in a given area. In other words, they live in a particular area or habitat. And therefore, the most basic quantitative property of a community is what's called species richness, the number of species that we're talking about, or some other predefined operational taxonomic unit, like genera or morphospecies or something like that. Okay, so richness. But there's also abundance. That's the number of individuals per species. Some species are common, meaning that they have lots of individuals, and other species are not. They're represented by very few individuals. And so one aspect of abundance is called evenness. That's the relative equitability of the distribution of individuals across species in a community. If every species in a community is represented by the same number of individuals, then that community is called perfectly even. On the other hand, if you've got a community where most individuals belong to one species, then that community is considered to be dominated by that species. And those two situations of evenness versus dominance are very different. These two properties together, richness plus evenness, are what characterize diversity or biodiversity. And then these species are living in an environment. The environment includes both abiotic and biotic factors that determine species occurrence and abundance. And so we'll be dealing with data for all of these this semester. Second question, why do we care? Why do we study ecological communities? Well, biodiversity losses are accelerating. We are in a biodiversity crisis. And so there's need to be able to quantitatively describe communities in order for conservation and management activities to occur. We need to be able to monitor changes in communities over time, to evaluate management actions conducted on their behalf, and to compare different regions. And so conservation biologists, wildlife biologists, natural resource managers, and community ecologists all need to have a quantitative set of skills to work on the biodiversity crisis. This course focuses on some, but by no means all, of these methods. Next, what kinds of data are involved? So a community is, by definition, a multi-species assemblage, with each species represented by different numbers of individuals. This collective exists in a discrete area that has environmental characteristics like soil type, or canopy cover, or soil moisture, or depth, or salinity, and so forth. And those environmental factors may limit what species can be present or what species can be present in what abundances. And so examining ecological communities necessitates multivariate data analyses. And so most communities at minimum involve two data sets. One is called a site by species data set, and the other is a site by environment data set. A site by species data set has data on species abundance or occurrence at various sites or sampling locations. These data can be binary, species presence absence, or they can be continuous, the abundance or the count of each species present. Then you may or may not also have measured some environmental variables at those same sampling locations. And so that's the site by environment matrix. So typically our focus is on whether there are any patterns of species abundances or occurrences by sites, whether sites show patterns in environmental variables, and whether species are associated with those environmental variables. However, before we can do any kinds of analyses on these data sets, we need to do some data screening first. We screen for zeros, and by zeros I mean species absences. That is not the same thing as missing data, and we'll deal with both of these in this class. Outliers are extreme values that can push an analysis or the conclusion that you draw from an analysis in a certain direction that may not represent the norm. And speaking of norm, normality, the assumption of normality, the normal distribution, statistical normality, is also something that we will be dealing with, or the lack thereof. And so we will be discussing data transformations, and in the notes actually for today I discussed data transformations that are needed for certain types of data in order to deal with matrices that are characterized by lots of zeros, to improve normality, or to regularize outliers. And so this week on Thursday we will have our first live session, and that's simply going to be a getting to know you kind of session, and experience in how the live Q&A format goes. Next Tuesday's lesson will be an introduction to R, RStudio, and R Markdown, and it'll also introduce you to some of the data sets that we will be using in this course. I'll see you then.